so today we're going to go a little bit over chapter 6 just briefly, but we are going to be focusing on chapter 7. So your aerobic, respiration, anaerobic, and fermentation will go in pretty good detail about glycolysis, Krebs, and etc. But first, I just want to start with a warm-up to get us all in the mindset of respiration. So my first question is, which of the following is the oxidized form and which is the reduced form out of these two, NAD plus and NADH? And anyone can speak up.
Isn't NADH when it takes an electron? So that's oxidized, correct?
So NADH, it has an extra electron, okay? So I always like to think of reduction as kind of the opposite of what you think, because you're thinking if you're reducing something, you're taking something from it, right? But it's actually the opposite. Reduction is adding an electron to something. So I always like to write NADH like NAD plus minus, because what is a hydrogen atom? Does anyone know, like, has anyone taken a chemistry class? This is like very, very basic chemistry, so I'm not, yeah, double gas. Okay, I'm just wanting to focus of what makes hydrogen hydrogen. Its atomic number is 1. What does that mean?
Means it has a proton and electron.
Exactly. That's all hydrogen is a proton and an electron. So sometimes when I'm looking at this, because you may be asked at some point, which is the oxidized form, NAD plus or NADH, I like just to write NADH as plus and minus for the hydrogen. And then I can look, okay, so the only difference between my NAD plus and my NADH, right, is the addition of this electron. So if NADH has an electron, is it oxidized or reduced if there's been the addition of an electron?
Reduced.
It's reduced. And another way you can think of this too, rather than thinking opposites, right, is if we're adding an electron to something, what's happening to its overall charge? So like, let's say something had a charge of 2 plus, and then I put an electron in there. Now it's plus. Yeah, it reduces the charge, right?
Okay. Yeah.
So whatever way rings true with you of like trying to remember, oh, typically I think reduced means taking, but since it's the opposite, now I know it means giving an electron. Or if you like to think, oh, we're reducing, by giving electrons, we're reducing the charge, right? However you want to think about it. So for this question.
So when something is being oxidized, it's essentially freeing up electrons?
Yeah, so if something's being oxidized, we're taking electrons, and we'll talk about that when we get to the Krebs cycle. But if we're oxidizing something, we are taking electrons from it.
Right.
Okay.
Okay.
And that's going to be important when we talk about respiration, because that's what the ETC depends on, is high energy electrons. And where do we get those electrons from? We oxidize glucose. Okay. And so just all I wrote here is oxidation is removing electrons. So that's going to be my NAD plus, right? Because it's missing an electron. So an electron has been removed from it. And then reduction is adding electrons, which would be my NADH because it has that electron there. So does everyone feel okay with oxidation versus reduction?
Yes.
Yes. Okay, great. All right, so now let's move on to the next question. And so I'll just have you guys think about this and answer, but there is something that I want to clarify a little bit when we talk about this explanation. Okay, so how would you classify sulfur oxidizing bacteria that resides in the bottom of Yellowstone National Park hot springs? So like if there's like hot springs at Yellowstone, this bacteria is like at the bottom of the hot spring and it oxidizes sulfur. Chemotroph. Okay. Yes, chemotroph. And why do you say chemotroph?
Organic.
Using an inorganic molecule. Okay, yes, that's going to be our next clarification. Okay, so we have chemotroph. What's on the other side of chemotroph? So, like, remember the table we had? Phototroph. Yes. Okay.
Right.
So we have chemotroph, and chemotroph is what they're getting energy from. Chemo. Yeah.
The breaking of chemical bonds.
The breakage of chemical bonds. Okay. And then phototroph, where do they get their energy from?
Sunlight.
Sunlight. Okay, so we know we're talking about a chemotroph here, okay, because our bacteria is at the bottom of this hot spring, so it probably won't be getting too much sunlight. So we're talking about a chemotroph. Now, somebody said using an inorganic molecule. Is it going to be a chemoorganotroph or a chemolithotroph?
Hmm. I'd say it's an what? Oh, organotroph. Well, if it's using inorganic, isn't it a lithotrope?
Lithotrope. Okay, let's leave it there for right now. So it's a chemolithotroph, yes. And then is it going to be a heterotroph or an autotroph?
Depends on if it's using oxygen or CO2.
Yes, it's a heterotroph. Okay. How
Okay.
I like to think about autotrophs is autotrophs, I like to think about glucose. If we have an autotroph, auto means self, right? So they're self-making glucose. In other words, photosynthesis, right? They can make their own food. That's an autotroph. Okay. Heterotroph, they're getting glucose from other sources because hetero means other. So they need to get their glucose from an autotroph, okay, because they can't make it themselves. So my classification for this was chemolithotrophic heterotroph. Now I want to go through one by one. I first said chemotroph because it is getting the energy from the breakage of chemical bonds, right, rather than sunlight. Here's my clarification for this one. So lithotrophy and organotrophy can be a little bit confusing, okay? And I struggle with this too. I was doing some research last night and something that I found. So I told you guys before that organotroph, right? They perform aerobic respiration and lithotroph is going to be anaerobic. It's a little bit more complicated than that. What the organo or litho is referring to, and here, maybe it's going to help if I go to this. Okay, so we have organotrophic or lithotroph. What that is referring to is where it's getting its electrons from. Okay, so process of getting electrons, is that oxidation or reduction? If we're taking electrons from something. It's right on the screen. If we're taking electrons from something, is it oxidation or reduction? Oxidation, okay? It's oxidation. And remember, this organism is a sulfur oxidizing bacteria. So where is it getting its electrons from? If we're oxidizing sulfur, yeah, we're getting the electrons from sulfur, okay? So this is why it'd be a lithotroph because it's oxidizing an inorganic molecule, okay? If we look at ourselves, we're organotrophs because we oxidize glucose, which we're going to talk about in detail during the session, to get our electrons, okay? It's a small nuance. I wouldn't really focus too much on that nuance between organotroph and lithotroph. The main thing that you guys should know, though, is lithotrophs get their energy from rocks or inorganic substances. Okay, you're most often going to see it termed as rocks because litho means rock in Greek or something like that. Yeah, either Greek or Latin. But you might see something along the lines of, if this organism gets its energy from rocks, what is it called? It's going to be a lithotroph. Okay. All right. And then I said heterotroph. Oop, I always do that. Heterotroph, because it does not perform photosynthesis and it's getting its glucose from other sources. Okay. All right. So do we feel okay with that question? Okay. All right, so let's move on to our first activity, which is going to be fermentation. So what does fermentation start with? Always.
Glucose.
Glucose, yes. And the process we're going to start with is called what?
Glycolysis.
Glycolysis. And what does glycolysis mean? Hold on.
Breaking of sugars.
Right? Because we have glyco, which is our glucose. Hold on. I'm going to write this a little bit differently. Glucose. And we have lysis. Sorry, bear with me. My pen's acting a little bit funny. I was in chemistry yesterday and we were using this really hot Bunsen burner. And I went to move something and I used my Apple Pencil to move it and it just like melted the tip. I know, big brain moment. But anyways, so bear with me. My pencil's a little bit funky today. Okay, what does lysis mean?
Break.
To break. So in glycolysis, what are we doing?
Breaking sugar.
We're breaking glucose. Okay, so this C6H12O6, that is my chemical formula for glucose. What I care to mention in my drawing is the 6-carbon chain. So 1, 2, 3, 4, 5, 6. So in glycolysis, we are going to break glucose. Okay, we're going to break glucose into something called G3P, right? And G3P, what the 3 is referring to is that it's a 3-carbon molecule. So we can see there we have literally split glucose in half, right? And in order to split glucose, what did we have to use?
ATP.
ATP. And how many did we use?
2.
2. Okay, so that's what you see here. I had 2 ATP. We used those ATP to split the glucose, and now I have 2 ADP. So let's go down to our accounting. In the glycolysis step of fermentation, what goes in? What did we start with?
Glucose.
We started with 1 glucose. 1 glucose. All right. And then what else did we start with? Two ATP. Okay. And we used those 2 ATP so that we could break the glucose in half. Okay. Two ATP. All right. So our next step here, we are going to actually oxidize G3P into pyruvic acid. Or pyruvic acid is also called pyruvate. They're like the same thing. Okay. So in our process of oxidizing G3P into pyruvate, what's going to happen? And remember
Take
sorry.
Take electrons.
We're going to take electrons. Does anybody know where we're going to put those electrons? NAD plus, okay? So we have NAD plus. What is it going to turn into when we give it an electron?
NADH.
NADH, okay? Oh, and that's the other thing. Hold on. Okay, so just focus on the NAD plus right now. We had NAD plus, which just exists in the cell cytoplasm. From this process of turning G3P into pyruvate, we get NADH. So we have taken an electron from G3P and given it to NADH. Okay. NADH is what's called our high energy electron carrier. Okay. All right. And what else are we going to get in this process? Two ATP. Exactly. Okay. So throughout this process, we're also going to be able to take a phosphate group from G3P and give it to ADP to make ATP. Okay. And so this is going to happen on the other side as well. Okay. I'll erase some stuff in a second. Okay, so this is basically glycolysis. All right. This is just the process of glycolysis. But right now we're talking about fermentation. And what is fermentation? It's glycolysis.
Over and over.
Over and over. It's literally just repeating glycolysis, okay? But in order for us to repeat glycolysis, what do we have to do? What did we make in this second step where we took G3P and turned it into pyruvate? We made NADH, right? What are we going to do with that NADH? It's holding on to some electrons that we don't necessarily need. Sorry, I think you're muted, Kristen. I can't hear. Oh, it's okay. Okay. But yes, in the chat, oxidize NADH and make lactic acid or ethanol. Exactly. So we're going to take an electron from NADH. So we are going to oxidize it. Hold on. Sometimes I never know like where I put my stuff, so I just have to. Okay. So we are going to take our NADH. Hold on. There we go. Okay. So we're going to take our NADH and we are going to oxidize it, meaning we take an electron from it. That electron is going to go to pyruvate, which will then convert pyruvate into lactate, or otherwise known as lactic acid. Okay. When we give that electron to pyruvate, the NADH is now going to be turned back into NAD plus so that the cycle can repeat. Okay. As long as there's more glucose, now we have our NADH again. It's been recycled. So now our whole cycle can complete itself again. Okay. So the other token, if we don't make lactic acid, what are we going to make instead?
Ethanol.
Ethanol. Okay. And this is a two-step process. And there's one more product we have in ethanol formation than we do in lactic acid formation. So again, we have this high energy electron carrier, and we need to find a way to convert it back to NAD plus, right? So where are we going to put our electron?
Pyruvate?
Yep, we're going to give it to pyruvate. And when we do this, it's going to form acetaldehyde and carbon dioxide as a byproduct, okay? So carbon dioxide is going to come out as a byproduct. And from our acetaldehyde, it's then going to be converted into ethanol, okay? And this is the step when the acetaldehyde is converted into ethanol. That's the step where our NADH drops off its electron, okay? So first what we're going to do is take pyruvate and we're going to convert it into acetaldehyde, and carbon dioxide is going to be released. Then our NADH is going to drop off its electron to the acetaldehyde, and it's going to become ethanol. And then our NAD plus is now regenerated, and the whole process can go over again. Okay. Does this make sense, kind of?
Yes.
Okay. This stuff, it does take a lot of practice, guys. Okay. So I know it's like super overwhelming at first when you see it all, but really what it is, you have to draw out these maps. This is going to help your understanding so much if you sit down and spend time to draw out these maps for yourself. But it's just practice. That's all it is, because it is a very confusing concept. Okay. All right, so now let's do our accounting. And I want you guys to understand too, I like split this graph in half, right? Just so I could show you on the same page lactic acid versus ethanol. If this was only lactic acid formation, this entire process would happen on the other side as well. Okay, so it would be doubled, but I just only showed you like one kind for each side. Does that make sense? Okay.
Mm-hmm.
So all the products that I have on this side are doubled. Okay.
Will you post this somewhere?
Oh yeah, for sure.
Okay. Awesome. Thank you.
So yes. So let's go back to our glycolysis. So we put in 1 glucose and 2 ATP. What did we get out of just the glycolysis part? Or this is the doesn't each only produce 2 ATP total per glucose molecule. Okay, so the word there, total, I want to focus on, okay? So we used 2 ATP to break glucose, and on each side we made 2 ATP. So what is our net gain of ATP?
2.
2, but our total ATP that we made is 4, okay? So the ATP we made, we made 4, but really we made 2 ATP. What else did we get out of glycolysis?
2 NADH.
2 NADH, and we see that here, right?
And then 2 pyruvate.
Exactly. So I have 1 NADH, 1 NADH there, 1 pyruvate, 1 pyruvate. So we have 2 NADH and 2 pyruvate.
I have a question. So on
Yeah.
our charts, it says glycolysis and like Krebs cycle. Fermentation doesn't really go through Krebs cycle, does it? Or is that no?
Nope.
Okay, so it's
Oh.
just glycolysis on that, and then
Yes.
okay.
So hold on, let me write this real quick. Okay. So fermentation is literally just glycolysis over and over.
Okay.
And the reason why we have lactic acid formation or ethanol formation is because we need to empty out the electrons from our high energy electron carrier. We need to get rid of that electron so that it can be converted back into NAD plus and the cycle can continue. Okay. That's the only reason that it makes lactic acid or ethanol.
Okay, so it's just glycolysis over and over and over, pretty much.
Exactly. And either lactic acid or ethanol plus CO2 is going to be byproducts.
Perfect.
Okay. And that's something I would remember as a difference between lactic acid fermentation and ethanol fermentation is that ethanol fermentation is also going to have a byproduct of carbon dioxide.
Okay.
And glycolysis is going to be the same for each respiration process that we go through. So like our first step of glycolysis where we get 2 pyruvate, 2 ATP, and 2 NADH, that is going to be the same process each time.
Okay.
Okay. All right, so now let's do our accounting for the lactic acid formation. What went in lactic acid fermentation? So our second step here, 1A.
Pyruvate.
Mm-hmm. And we do understand we had 2 pyruvate, right?
Right.
And do we understand where I'm getting the 2 from, right? Because this whole process would be repeated over here as well. I just wanted to show you both on the same chart. Okay. And then, hold on. In fermentation, are the final electron acceptors organic or inorganic molecules? In fermentation, they're organic. Yeah. Yeah. And when I post the recording, Kristen, I know you came a little late. Look over my explanation to question number 2 because I clarified a little bit more the organotrophic versus lithotrophic. Okay. Because I read some stuff on it last night that like clarified some other things for me. Okay. All right. So we had 2 pyruvate go in, and then what came out for lactic acid fermentation?
2 lactic acid or lactate.
2 lactates. And I guess you could say that another product would be, right, like our recycling of NADH into NAD+. Yes. The 2 ATP is actually just for glycolysis. Okay. So I'm in this accounting, I'm just talking about the lactic acid formation step. Our 2 ATP is just glycolysis. Okay. All right. So then for ethanol formation, what went in?
2 pyruvate.
Yes, 2
Two
pyruvate.
pyruvate.
And what came out?
2 ethanol.
Yes.
Oh, we're doing, okay, that makes sense.
No, you were saying the right thing.
Well, with the extra steps, you get CO2 and acetyl. That one.
Yeah, I'm just talking about end products. Okay, so our end products are ethanol and CO2.
Oh, okay.
The acetaldehyde is kind of an intermediary. I can't even say that. It's an in-between step. Okay.
And that CO2 is like an ethanol formation, so that's like what yeast does, right? And that's why bread rises because of
exactly.
CO2. Okay.
Yeah, and that's why too. Like people who make their own alcohol or whatnot, you need to open the lid just a little bit because it's going to start releasing carbon dioxide, and you don't want the thing to like explode.
Well,
right?
They have special caps that have like a floater that lets it bubble. You know.
I don't know. But same idea.
Yeah.
All right, so do we feel okay with fermentation?
Yeah.
Okay, so if you were to be asked possibly, what are the end products of alcoholic fermentation? You could answer that.
CO2 and ethanol.
Yes. Okay, great. Okay, so how many ATP did we make total through lactic acid fermentation? 2.
Oh, that's just the...
Right, because it only came from glycolysis. We didn't make any ATP in lactic acid. I'm just talking about the whole process together. We made 2.
Yeah.
Now for ethanol fermentation.
2.
Okay. So now let's move on over to our second activity, which will be aerobic and anaerobic fermentation, or respiration, not fermentation, aerobic and anaerobic respiration. So just to like give you a breakdown of how I set this up, in the middle, we're going to put all of our steps, but on the left-hand side, I wanted to talk specifically about aerobic and eukaryotes so that you can get the different structures, right, of like mitochondria and the mitochondrial inner membrane, which we'll get to. And then the right-hand side, I wanted to focus that on anaerobic respiration, which, where does that only occur?
In prokaryotes.
In prokaryotes, right? So this will be all of your prokaryote structures. But again, like before, it's just important to understand if we were talking about aerobic, it could happen in prokaryotes too. So it would be the structures on the right-hand side. But I just wanted to be able to put everything in a concise place. Okay. Great. All right. So we're going to start with our process of glycolysis. What does it start with? Glucose. Okay. And remember, any form of cellular respiration starts with glycolysis. So we have our glucose and we're going to break it, right? And to do so, we need to use how many ATP? 2. So we're going to break it into G3P. What are we going to convert G3P into?
Pyruvate.
Yes. Dear God. Okay. No. Ah. See, what I do is I draw all of it and then I white it out. And then I try to erase what I've whited out, but I don't know where it is. So I just have to like guess. There it is. There's one pyruvate. Ah. Another pyruvate. Okay, so when we turn G3P into pyruvate, we are oxidizing the G3P into pyruvate. So what do we get?
NADH.
NADH, right? Because we took an electron from G3P, gave it to NAD+, and now it's NADH. This is going to be on both sides. Oh, and other thing, I always put a box around everything that's a final product in the process. Okay. All right, and then what's the other thing we made when we turned G3P into pyruvate?
2 ATP.
2 ATP on each side, which gets us 4 ATP total. Okay. Okay. All right. So does everyone feel confident about glycolysis? Great. Now, this drawing I have is to show you where this happens in a cell. So in a eukaryote, where is this going to happen?
Cytoplasm.
In the cytoplasm. So like this is the cell membrane, right? And then this is the cytoplasm. Okay. And in a prokaryote, where does it happen?
Cytoplasm.
In the cytoplasm. Okay, so now what's our second step going to be? I called this 2A. I put prep step and Krebs together. So 2A. Pyruvate processing. What do we do in pyruvate processing? What are we trying to turn pyruvate into?
NADH, right?
Yes. We get an
Or
NADH. So
we're oxidizing it to
Mhm.
form acetyl-CoA and NADH.
Yes. And there's one other thing we make.
CO2.
CO2. Okay, so this is my acetyl-CoA. We've taken a carbon group off. And now that carbon group is released as carbon dioxide. Okay, so that's our other end product of pyruvate processing. Okay, so I get 2 NADH and 2 CO2, right? Because it happens on both sides. Both sides. Okay, so now I have my acetyl-CoA. And what is my acetyl-CoA going to go to?
Mitochondria?
If we're in a eukaryote.
Okay.
And if we're in a prokaryote, where is this going to happen?
In the cytoplasm.
In the cytoplasm. Okay, so the Krebs cycle.
Faith, the prep step doesn't happen in the mitochondria?
The prep step.
Anjali had them like together.
Yes, it does happen in the mitochondria. So if we're talking about eukaryotes, it happens in the mitochondria, and I'll tell you the specific place in the mitochondria. I was just thinking if it happens in the cytoplasm and is transferred there. It happens in the mitochondria. Our pyruvate is transferred to the mitochondria, then the prep step occurs and the Krebs cycle occurs. In prokaryotes, it all happens in the cytoplasm, right? Because we don't have mitochondria in prokaryotes. Okay, good. Okay, so my question to you, what is our main purpose of the Krebs cycle?
To get more NADH.
Yeah, and so if we're getting more NADH, what are we doing? What process?
Oxidizing things and reducing the high electron carriers.
Yes, so to get NADH, we're oxidizing. So the Krebs cycle itself can be a pretty complex cycle, okay? What we care about, I don't care about like all the in-between steps. We want the just end products of the Krebs cycle, okay? So, like you said, we're going to produce a lot of NADH. Hold on, I'll just put all of the... Okay. So yes, we're going to produce from 1 acetyl-CoA, we get 3 NADH, 1 FADH2, 2 CO2, and 1 ATP. Okay, I put all of my high energy electron carriers in blue. Okay. Yes, and it's doubled again, right? So that was for 1 acetyl-CoA. We have 2 of those that come from 1 glucose molecule. Okay, so all of this is doubled. That's what I have on the other side. Okay, so one thing I want to clarify here is where does this take place in the mitochondria? Okay, so this is my mitochondria. The middle of the mitochondria is called the matrix. And this is filled with systole, which is just like cytoplasm, okay? And so one interesting thing for anyone who cares, what does the mitochondria on its own kind of look like? A bacteria, right? I think that this is pretty cool. The mitochondria, their structure is very similar to that of bacteria. And it's actually evidence that bacteria evolved into eukaryotes, right? But anywho, so the middle of our mitochondria is called the matrix, and this is the systole. And this is where the Krebs cycle takes place. So I'll just draw this as like the Krebs cycle, since it's a cycle. That's where this is all taking place in the mitochondria, okay? Now, when we get to the ETC, where it's going to take place is in these little folds of the mitochondria, okay? So like this is going to be called our cristae. And that's where the ETC is going to take place, and we'll talk about that in just a second, okay? Of why it takes place in these folds.
So Krebs, matrix,
Mm-hmm.
ETC, cristae.
Exactly.
In eukaryotes.
Yes. And then the Krebs cycle for prokaryotes, where is it all going to take place?
Cytoplasm.
The cytoplasm. Okay, so now let's go back to our products. I have now done my job, and I've fully oxidized my glucose. Now, through this whole process, we've oxidized glucose fully, and now we have our high energy electron carriers. One thing I want to point out is for aerobic respiration, we get complete oxidation. Okay. And for anaerobic respiration, we have incomplete oxidation. So meaning all the electrons we could have taken out of glucose in anaerobic respiration, we weren't able to take out the maximum number of electrons. So it's incomplete oxidation, which is then going to result in less ATP made, okay? The details of this, I don't understand. I don't really think there's much like research on this or like concrete knowledge. It's hard for me to find anything on it. But in terms for like what you guys need to know, in aerobic, you get complete oxidation of glucose in the Krebs cycle. For anaerobic, you get incomplete oxidation of glucose in the Krebs cycle. Okay, so we'll just leave it there for now. Good. Okay, so we have our high energy electron carriers. Where are they all going to go?
To the cristae.
To
Yes.
the ETC.
To the ETC. Okay, the cristae if we're in the mitochondria. And so what's going to happen here is I have my high energy electron carrier, NADH, right? And I can also write this as just NAD plus minus, right? He is going to drop off his electron. Woo. I was fighting with this before the session. I was trying to get it to work. Okay. So he's going to drop off his electron to a carrier molecule that's in this inner membrane of the mitochondrial cristae. Okay. And so when he drops off his hydrogen here, this hydrogen is going to be transported through this membrane. And I'm not hydrogen, electron. I'm sorry. The electron is going to be transported through the membrane. The electron is negatively charged, so what is it going to attract? Positively charged ions. Okay, this is the other thing. In this mitochondrial matrix, so the systole fluid, there's free-floating hydrogen protons. Okay, so like in the cytoplasm and the systole, there's some free-floating protons. So what's going to happen is when we have this electron that's on the electron carrier, when he's on this side, oh my gosh, hold on. When he's on this side of the inner membrane of the mitochondria, he's going to attract these hydrogens to him. But since he's being transported, you know, he's like on this roller coaster ride, when he gets up here, he's going to push the proton out. He's going to push the proton out, okay? And this cycle is going to continue to happen. He's going to keep pushing protons out, okay? Until we have more protons on the outside of the membrane than we do on the inside, okay? And now what has my NADH become? NAD plus. So he's been recycled. My NADH is now NAD plus, and he can go start the whole process over, right? So I have my proton gradient. How do gradients work?
They go from high concentrate, go in.
Yeah, they go from high to low. So now I have a high concentration of protons on the outside, but protons can't easily diffuse through the membrane. So they're going to have to go through a special channel protein that is called, does anyone know the name of this?
ATP synthase.
Exactly. ATP synthase. Sorry, I can't write it all at once. What does the ASE mean?
The enzyme.
Yeah.
It's an enzyme. So when my proton comes out or goes through ATP synthase, what's going to come out is
ATP.
ATP. Okay, so my electron has gone through his whole roller coaster ride, and now he's over here. And my proton has gone through ATP synthase, and now my proton's over there. Okay. I need something to be my final electron acceptor, meaning we have this free electron out here, and we have to have something to absorb it because we can't just have a free electron floating around the cell. So what are we going to have take up this electron?
Oxygen.
Oxygen. So we're going to have our oxygen here. These are going to come together. So it's a little bit complicated of a process, but we're going to get rid of this proton, get rid of this electron. This becomes hydrogen. We have an oxygen, and we have, this happens frequently. So then we're now going to get another hydrogen. And now what have we made?
Water.
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 Okay. So water is a byproduct of the electron transport chain. Okay. So now let's go to the other side of prokaryotes. And in my prokaryote, I'm going to talk about anaerobic respiration. So I have again my NA.
On gram-negative bacteria, does this happen in the inner membrane or the outer membrane?
This happens in the inner membrane. I'm fairly certain. You won't be asked that question of where it happens, but I'm pretty certain it's in the inner membrane. So that's why I just gave here, this is like kind of a generic bacteria. I'm just showing inner membrane and then a relatively small amount of peptidoglycan. Okay. So I have
Sorry, on piggybacking on that question, it's like it happens in the inner membrane because it starts in the cytoplasm, so it can't technically get through to the outer one, right?
Yeah. Mm.
I think that's what we were kind of thinking.
Yeah,
so, yeah,
you're right with that. But
okay,
for any bacterial cell, we're just going to say it's going to happen at the cell membrane, which is the inner membrane. Okay.
sounds good.
It's going to be the same process. I'm going to have NADH. And instead of it being in the mitochondria membrane, it's just going to be in the cell membrane. Okay, so same exact process. I can write my NADH as NAD plus minus. My electron is going to go onto a carrier protein, which will then take him for a roller coaster ride. Up and down, up and down, up and down. He's going to come out the other side. And when he goes through this roller coaster ride, he's going to be picking up protons from down here and pushing them out here. Right? And this proton is going to want to come back in. What structure does it have to go in through?
ATP synthase.
ATP synthase. Okay. And what's going to come out?
ATP.
ATP. Okay, so let's say, hold on. So here's my electron. I have my free electron at the end. This time I'm talking about anaerobic respiration. So is oxygen going to be our final electron acceptor? No. Instead, the example I'm going to use is just going to be sulfur. Okay, so if I have sulfur, I have my proton came through the ATP synthase as well. So now he's over here. These are going to come together to form hydrogen. And let's say I get 2 of those. Now I'm going to have hydrogen sulfide. Okay. So it's pretty much just, it's the same exact process. Your end product is just different. Okay.
And less ATP, right?
Less ATP because there's less high energy electron carriers because glucose couldn't be oxidized all the way. Okay.
And no CO2, right? In the steps above it.
CO2.
Yes.
Yes, CO2.
There will be CO2.
Oh.
Okay. Because CO2 isn't a, so like oxygen down here, we're talking about atomic oxygen, which is a radical form. And so if a cell is using atomic oxygen as its final electron acceptor, it has to have enzymes to break down that oxygen if it turns into hydrogen peroxide or, just a radical form of oxygen. CO2 is not radical, so anaerobic organisms are fine with it. Okay. Because the CO2 comes out from glucose.
So then would your end product still be the same for Krebs cycle as aerobic?
Yeah, so that's the thing where it's like I couldn't find too much information on it. I don't know exactly, how less of NADH are produced. That's kind of not clear. in a lot of stuff. It's hard to find information on anaerobic respiration for whatever reason. But like I said, all you need to know is oxidation is not complete in the Krebs cycle. For the purposes of this class, we're going to keep all of the end products the same, except for the amount of ATP produced at the end. Okay. And the amount of ATP that the amount of ATP that each NADH and FADH2 can make is also a variable. It depends on other things. So we're not going to focus directly on, this many NADH makes this many ATP. We just care about the overall process. Okay. All right. So do we feel okay with all of this?
Mm-hmm.
Did it like clear up some things? You feel a little more... Okay, great. Like I said, it's a very confusing subject in general, so it just takes time, but I've tried like reducing it down to its bare bones. Okay, so let's just do our accounting because this is a really helpful process in understanding all of this. So for glycolysis, what goes in?
Glucose.
1 glucose. And what else goes in?
2 ATP.
2 ATP. What comes out?
4 ATP, 2 net.
What else?
2 pyruvate.
2 NADH.
Mm-hmm.
2 NADH. Okay. For the Krebs cycle, what goes in? Or the prep step of the Krebs cycle, I'm sorry.
Pyruvate.
So our pyruvate from above will go into there. And then what do we get out?
4 CO, or wait. 2 NADH.
And 2 CO2. And 2 acetyl-CoA.
Yes.
I have a question about acetyl-CoA. For the processing, it takes 2 pyruvate plus 2 CoA. Are those just in the mitochondria hanging out?
Yeah, that's a coenzyme. So yeah, that's just going to be that's just going to be in the cytoplasm or the matrix of the mitochondria. A lot of these materials like our NAD plus or ADP, the CoA, that's already like just floating around in the cell. The cell is like making those already, just like waiting for this process to happen. Okay.
Okay.
So, we get 2 NADH, 2 CO2. Okay. And then in our, I put prep step and Krebs together. Okay. That's why I was like, why did I have, hold on. Okay. So how many NADH total do we get out of this whole thing? Hold on.
6.
Hold on. Okay, we get 8 NADH because I'm talking about the whole thing together. So this is 2A and 2B together. Okay, so I get 2 NADH from 2A, the prep step, and then I get 6 NADH from 2B. So altogether, we get 8 NADH. Okay. And then what's the other thing I get?
FADH2.
FADH2, and how many?
2.
Yes. And then I also get 6 carbon dioxide and 2 ATP. Okay.
Mm-hmm.
All right. So now if I'm talking about aerobic respiration, what goes in to the ETC?
NADH and FADH2.
How many of each?
Yeah. 8 and 2.
Yeah. So 8 NADH. Come on.
Because everything is fully oxidized.
Yeah. Hold on. Wait 1 second. It's not 8 NADH. It's 10 because look where we had this NADH as well from glycolysis. Okay.
Oh.
So all of our high energy electron carriers go to the ETC. Okay, so it's going to be 10 NADH total and 2 FADH2. What comes out?
ATP.
Yes.
36 ATP? Or
Yeah,
30?
the number kind of varies. Especially if we're talking like prokaryote versus eukaryote, the number tends to vary a little bit. And the reason why in aerobic respiration, eukaryotes are going to produce less ATP than prokaryotes is because they have to take this extra step of moving the pyruvate, right, into the mitochondria. And so it's going to take energy to move that there. Okay. So that's where we lose a little bit of ATP production in eukaryotic aerobic respiration, but that's just a very small detail that you guys don't really need to know as much. So out of this whole process, roughly, just what we're looking at here, how many ATP did we make in aerobic respiration?
34, 32, 34.
Yes.
And water.
Yes, water is the other one that I didn't put on there. I think it's 6 H2O that they make.
Yes.
Yeah.
It is.
Yeah. So we'll put that on there. Okay. So then let's look at our anaerobic side. What are we going to have go in? And remember, this is just like total. We don't have to get into all the nuances of incomplete oxidation yet. We're just saying generally.
I'll say from Cribs.
So for the, 10 ADH and 2 FADH2. Yeah, so for this one, I just put the same amount, okay? The only place where I change my amount is with the ATP produced.
Okay.
Because like I said, there's really like not any good information of exactly how many NADH and FADH2 are in anaerobic. So I'd rather just use this number that I know you guys should probably know. Okay. And then what do we get out?
2 to 32 ATP?
Yeah, so the 2, what it's saying there is fermentation. Okay. So that's what I mean. Like, there's not good information about anaerobic respiration. And this, I think I put, hold on.
That 2 to 32 is from Anjali. She said that the anaerobic can produce anywhere from 2 to 32 depending on the carriers because they're just not as good as oxygen.
Okay.
So that's like where we're getting our 2 to 32.
Yeah. If you search it up on Google, it also says 2 to 32. And I just have a feeling the 2 is referring to fermentation, and then because fermentation is technically anaerobic respiration, right? But then if you have anything above 2, then we're talking about, okay, you're using Krebs and ETC. And yes, final electron acceptors are going to vary, and that's going to change some ATP production. But anyways, so I just put here 28 ATP. So then altogether, how many do we get?
38 is 32.
32. Yeah. Okay.
Maths.
So then this last page here is just to like, it's just another chart. I like charts with this. We did this kind of in my last session, but it's just for you guys to like rewrite down all the differences, all the similarities. It's important to know anaerobic produces just a little bit less ATP than aerobic, and fermentation produces the least amount of ATP of all the 3. And yeah, and then where the processes take place in eukaryotes versus prokaryotes, which you should have all your info here.
Nice.
Okay. So that's what I got for today. I hope that was useful for you guys. I'll post this after the session. So.
Okay.
Excellent. Thank you.
Yes. Goodbye,
Thanks.
everyone. If anyone has questions, I'll stay on for a few more minutes. So,
I think we're good. Thank you, Faith.
yes, you're
Thank you.
welcome. Yes.
Thanks, Faith.
Yeah, no problem.
And your other study session is Sunday?
Oh, yeah, that's what I forgot to mention.
Sorry.
I'm having an exam 2 review session on Sunday. I put an announcement about it too. I'll make another announcement. So
Time, just so my brain hears it.
12 p.m. to 2 p.m.
Okay, perfect. Thank you.
Yeah, no problem. All right. Okay. So fermentation is only glycolysis. Okay. There's anaerobic respiration can involve all 3 processes, right? So like glycolysis, Krebs cycle, and ETC. The only difference is that the final electron acceptor will not be oxygen. It's just going to be some other inorganic molecule. Okay. But the only process that is just glycolysis over and over is fermentation. Okay. So we have 2 different kinds of anaerobic respiration. We have fermentation, which both prokaryotes and eukaryotes can do. And then we have the, anaerobic with glycolysis, Krebs, and ETC, which is for only prokaryotes. Okay. Yes. Okay. Bye.